it is a pleasure to talk to my friends and colleagues in Bergen, a place where I spent a decent part of my life, a pleasurable one, I must say. And I brought from there also a souvenir who became my wife over the time. So it is always when I see her, it reminds me of Bergen. Also, it is important to mention that this work actually has started in Bergen next to our rather focused research on ultrasound visualization. We also looked into visualization of molecular structures. The beginning was a push from our colleague Uwe Lampe, who has done first work in this domain from the visualization group at the very beginning. And then with others, most notably Julius Parulek, we have advanced the molecular visualization field further also into the direction of visual analysis of molecular structures. And this scope to a strong degree is at the University of Bergen with a slightly perhaps different scope focus than what we are looking at in the group back in Vienna and also at KAUST these days. So I will be talking about our recent works, which are related to 3D modeling and 3D visualization of biological ultrastructure. It is based on our prior work. So we somehow help ourselves in research by building on top of previous techniques which we have developed. And the one work which we are building upon is molecular rendering, which we have developed in the past in TU Wien, and molecular visualization. Here you can see just a benchmarking visualization, an ugly one, but which is showing you 15 billions of atoms which are in the scene, not effectively rendered. And that's the reason why we can achieve very high interactive frame rates. And this technology is necessary to have if we want to perform modeling and further visualization of models that contain a lot of atoms. But let me go straight to the scope of today's talk. And that is, you are not seeing all of my screen. I see. Is that?
On the bottom right, there is maybe a Zoom interface in front of your.
Yes, but it's there is not much content there.
Well, if it doesn't hurt, it's fine. I just thought because you have nice videos and everything, but don't you mind? I just thought
okay.
it's a one.
Even but if you turn off the screen sharing and turn on again, and not just share the PowerPoint window, but the entire screen, then that should be gone.
Okay, I will try that. I will try that.
I'm sorry for interrupting.
No problem. I think it's good to have it, but I have to optimize for video clips, and that might be actually a problem. But I will try now to have it as Stefan suggested. Now this is the whole screen, and I don't know if it's better, but I still see it here.
Yes.
Okay, I will keep it this way. It is just occluding this header, but I hope this is fine. So we have developed in the recent year or so a 3D modeling technique, which is fitting into the context of modeling biological ultrastructure in such a way that we are building on the previous literature of procedural modeling. However, the procedural modeling approaches previously were mostly dealing with text-based input, where you define your grammar, and then according to that, a certain set of buildings or other structures are being built. In our case, we are replacing this text-based form of input with a 3D modeling input, where you perform as a modeler certain kind of visual programming. We can formulate our our system as a grammar, which consists of non-terminal symbols, terminal symbols, some production rules, and initialization, where the non-terminal symbols will be geometric proxies, as can be seen here. Some geometric elements which will be eventually not visible in the final model of a certain structure. Then we have the terminal symbols, like, for example, this protein, which is assembling according to certain rules. The rules you can see here, for example, where I would define that the rule will be that this protein has to be at a certain distance from my geometric proxy, from these skeletons, as we call it. And if we change this skeleton from a line into a triangle, which is here, we can see that suddenly this rule is then reapplied, and it generates a totally different structure. We can have not only one distance defined, but several, and also our rule sets are distribution based. So we can, if we have several rules to choose, we can have a probability on a certain rule to be applied, and also the transformations do not need to be exact. So a particular distance, rotation, orientation can be can have a certain distribution. Like, for example, here we can see that there are two distances defined with a certain probability distribution, which is then reflected here in this rule execution. If we just switch this triangle with a point skeleton or point geometric proxy, then we have, again, a totally different structure, but adhering to the same rules. The termination of such a modeling process is finished by a certain set of iterations, or if we have structures like, for example, bilipid membrane, where we do not have an exact amount of structures, then we are defining the termination through the spatial availability. So if we have, if we want to place a certain object, and this object is placed several times unsuccessfully due to a collision conflict, then after certain amounts of such events, we will stop the population. The whole rule-based modeling is organized into hierarchy. So we can have one rule defined, and this rule is then placed in a different set of rules. And this way, we can hierarchically build up a certain ultrastructure. I would like to exemplify it on the case of the SARS-CoV-2, where this is, which is shown here on this illustration of David Goodsell. Here you can see a lot of the life cycle, in fact, of the SARS-CoV-2, but we will be focusing only on the mature virus, which is already budded from the host cell. You can see a lot of other structures here, how the production of the virus is performed by making some kind of a bulge out of the endoplasmic reticulum. There is a lot of viral RNA being duplicated, which is then packaged and condensated by these nucleocapsid protein systems into a budding virus that is then, after budding, is ready to infect another cell. So here, while the genome is rather long of this virus, we have only few of the building elements of the mature virus. Most notably the spike protein, which you have many heard of, and then we have other two proteins anchored to the membrane, the envelope protein and the membrane protein. And inside, in the lumen of the virus, we have the nucleocapsid protein complex, which is holding or condensing the viral RNA. And this, how it is exactly holding it, it's still subject of biological research. However, we can explore several hypotheses how this is done in our 3D modeling tool. It is important to mention that while the genome of the SARS-CoV-2 is 29,000 bases long, there are 3 or 4 virus proteins, which are actually forming the budded or mature viral particle. The other viral proteins are within the host cell, organizing the replication of the virus. So if we want to model a virus from scratch, we will really have to start from the very first elements. And the first element which we will build will be the envelope, the bilipid membrane. And the rules here will be that we will have one phospholipid here as a structure, and we will define the positioning of its siblings. So the neighboring lipids which are placed around it. We will define several rules around this lipid where its siblings can be positioned. Then we can also define a certain rotational variance distribution, so that not all these lipids are standing under the same orientation. So we have the chance to perturb the positions and the orientations a little bit. Once we have the envelope, well, one side of it, we can then replicate it just by mirroring the envelope, the single lipid membrane part to a bilipid membrane part, and then we are done. For the spike protein, we can simply define a so-called distance rule, where we have a certain pivot position of this molecular structure, this protein, and this pivot should be at a certain distance and at certain orientation from a certain skeletal object. In our case, this will be a triangle mesh. For the internal part, there have been several hypotheses, as I said. One hypothesis, which was at the beginning, the more prevalent one, was that the nucleocapsid protein complex is organized into kind of a rope structure, as you can see here. And it is based on the CTD and NTD domains, the carbon terminal domain and the nitrogen terminal domains, subdomains of this so-called N protein. And it is built as, this is the one dimer of this CTD, and with a so-called parent-child rule, which is a relative rule, we can simply mirror this positioning of the CTD into a dimer of the dimer, and then we can create even an octamer of the CTD. And this is a one building block along this chain, which is then folded inside the viral particle. On top of this chain, we have the NTD domains, and the NTD domains contain the binding sites for the RNA. So those will be holding and organizing the viral RNA and protecting it. We also need to model RNA, and I have to say that our system is not aware of any biology. All these rules are geometric rules, which we have to express from the very beginning. However, once we develop a certain rule, like for example, how an RNA is organized, then we can transform this RNA into any other system. And the rule set will be just executed as it was executed on another model where it came from. So here you can see how we generated the RNA string, where we have one of these line skeletons, and along these line skeletons we have a winding sugar backbone, on which from a certain distance of this sugar backbone there are the bases of the RNA. And we are defining this on a certain line. However, then we can transfer this rule on an arbitrary 3D curve. The 3D curve we are generating with a so-called connection rule, which based on a certain amount of binding spots, there is a curve created which connects most of these binding spots in one long 3D spatial curve. And then we are simply transferring this the line skeleton, or replacing the line skeleton with this complex curve, and populating the RNA on this 3D curve. And we can do it even in such a way that if we provide the accurate order of the bases, we can have a model which has a representation of the RNA accurate to the genome of the virus. So I will not show you this video because I will make an attempt to model the virus myself. Maybe, hopefully this will work on this laptop, so bear with me. So I'm starting one application, which is called Mesocraft, which is our 3D modeling tool. And I just want to show you how this approximately works. So we can just start with the triangle skeleton, and we will add the triangle into the scene. Now, once we have this triangle into the scene, we will add, let's say, a phospholipid. And we have it here. And what we can create now is a certain rule, simplest rule, and the only rule which I will be using in this demo after I need to highlight it. Okay, so it looks like I cannot highlight this guy now for some reason. Okay, so I can take him a little bit above this plane, above the triangle plane, and create a rule, parent-child, where the parent is the triangle and the child is the phospholipid. And once I create, oops, I have to select it again. And once I create this rule, I even take this guy away now. If I apply this rule, I suddenly have several of these phospholipids present here. Now I need to change several things here because first of all, I don't want to have too many of these elements present. Currently, it was set to 0 says fill until there is space. Also, I need to change the collision handling a little bit so that we can have some denser placements, less conservative one. And also, I will change that all these phospholipids can rotate arbitrarily around itself, and that also there is a certain allowed pitch and roll. Also, the distances, I can define that there is a certain distribution from the distances from our triangle. So you can see here how I created relatively quickly a membrane, and on this membrane, or one layer of the bilipid membrane, I can now put another type of rules of adding the spike protein and other proteins, but I will not do it because I think this will take too long time then. And I just, you can see that if I will continue with this video here, you will have a bilipid membrane, and then after a short time, you can see that we can add also the envelope protein and the other membrane protein, and we can create a triangle patch of an envelope, which then, if we replace this triangle with a triangle mesh, the rules will be executed on the triangle mesh instead, and we will have the complete envelope generated instead of just one single triangle. This modeling tool you can find on our publication website, which is nanovis.org, and it is the paper is called Modeling in the Time of COVID-19. There is the latest executable for Windows for NVIDIA graphics cards available for download. You can play around with it, and if you will do so and you will encounter certain difficulties, just contact me and I will be glad to help you in modeling whatever structures you would like to do. Currently, we are using this modeling system for modeling certain structures for our collaborators. And also we have some student projects where enthusiastic high school biology students are modeling some structures, like, for example, the T4 bacteriophage over this summer. So I hope next time we will be talking to each other, I can already show you a more complex model of the T4 bacteriophage. This modeling tool has as a byproduct the first 3D model of the SARS-CoV-2 variant, which not only was modeled from the envelope, but also the internal structures. And shortly after this medialization, we have been contacted by Tsinghua University biologist Sai Li, who said that he has a different hypothesis on how the nucleocapsid organization is inside the viral lumen. And here you can see his organization, where the nucleocapsid proteins are organized into some kind of a G structures, and these are then condensing the viral RNA. So, which was in some sense a proof that we can use our modeling tool in quickly updating the model. Also, you can see here that the viral spike proteins are no longer of one type, but there are some types which are called the open conformation spike protein or the post-fusion spike protein, as shown here. And they are distributed according to the latest knowledge from the biological literature. Sorry. So, but with this collaborator, Sai Li, we not only gained insight on how this nucleocapsid protein complex is organized, but according to him, but also he was providing us with the experimental evidence captured by the imaging he has provided. And the imaging is the Cryo-EM tomography, where the several Cryo-EM projections are then used for creating a tomographic reconstruction and a 3D volume. And here you can see, if you are performing certain image processing operators, how we can see the structures inside such a modality. And this detail, you can already see that there are certain viral particles here. You can see also a lot of noise around it, but you can also see some hints of spikes and these type of structures. So there is something in there, but still the quality of the image is, I would say, especially to Odd Helge, even more challenging than 3D ultrasound. But nevertheless, even for such a modality, we were thinking of how can we perform the 3D visualization directly out of such data. And we came up with a pipeline which involves the most popular technology of today, deep learning, in the specification or the classification process. You can see here that with this approach, we can effectively reduce the presence of the background of this salty solution in which the viral particles have been embedded in. And even more, we can segment various structures, some envelope structures, some lumen parts which are enclosed by these envelopes, and also some membrane-bound proteins like these spikes here. So, and this work is based on a very difficult to read 2D slices, as you can see on the very left. In the next to it, you will see the straightforward visualization of such a data, which will be just a clutter dominated by the background. And with this deep learning-based classification, we can extract the viral particles and even have a 4-component classification where we can assign different colors to different structures. How this is performed, we have been given very generously by Sai Li access to 300 reconstructed volumes. And out of these 300 volumes, we have used elastic segmentation tool, which is a very popular segmentation tool these days, for manual or semi-automatic segmentation of 60 of the best volumes which were in these data sets. Just a second, I will look into the chat. Okay, this is. And then after we perform these segmentations, we end up with smooth labels, meaning that some structures are given a probability whether they are part of a background or of a membrane-bound protein or of an envelope. And it turns out that this soft specification is perfect for a classification for the purposes of 3D visualization. Maybe not so much for other purposes, but this non-binary soft representation of classification is very suitable. So in fact, the outcome of a probabilistic segmentation is very useful. However, we always, in our first iteration, needed to do semi-automatic segmentation, which was working well for that one particular volume which we segmented, but was not really transferable across other volumes. For that, we have performed training of the so-called 3D unit network, which we then use also for inference when we are getting new data from cryo tomography. And using this inference, we obtain soft segmentation as the basis for classification, which is then used for automating transfer function estimation, which is a simple opacity ramp, which we estimate the basic parameters of. And the user can then adjust it or assign different colors to it. But basically, there is no user interaction in this volume visualization pipeline. Not so much, very lightweight. No transfer function design, 2D transfer function design, or other complex things. However, such a representation likely will then work only for the Cryo-ET data, and more specifically for Cryo-ET imaging, which contains viral specimen. I have here some comparisons of this process. Maybe these are details, but I just wanted to show you that the deep learning actually performed really overall much better, even compared to the semi-automated segmentation, where we can see that more of the structures have been pulled out. And the collaborators' satisfaction was especially in the fact that the segmentation works quite well in the so-called missing patch area, as these projections which are being created are only taken from certain angles. There are certain angles which are not covered, and these are forming this missing wedge, which is a missing information in the frequency domain, which then makes it difficult for example, manual segmentation of spikes, because they are very blurry, as is done by the domain collaborator. However, our system was able to actually pull out the presence of spike proteins also out of these missing wedge areas quite successfully. You can see here how we apply various illumination techniques. So this is, there is no shading. We are using then ambient occlusion, and then also soft shadows for further strengthening the 3D perception of the scene. If you see that all these viruses are somehow opened, that we can see inside this blue lumen, that is actually because of this missing wedge artifact, and we would need to perform additional research on making sure that this missing wedge is not manifested itself in the visualization. Our renderings, you might have be able to see our collaborators from Nanographics have made nice videos and nice images out of this technique, which have been then seen all around the world. Here is one video which has been seen by 150,000 people from this work. So if you have seen something similar like this, this was done recently by our group. But switching the gears a little bit, I would like to talk about one possible application of this generation of these models, and out of also this type of volumetric data, which I will come at the very end to, where once we have such a model, one way what we can do with it, and we will be performing experiments with this, is a simulation of such models which we generate. So we will create, so to say, the starting condition for performing molecular dynamic simulations. And the second application, which I'm always close to, is explanatory visualization of the models which we generate. What you have seen here was our earlier work where the user can rotate around a viral particle, the HIV, and can explore various structures. By this simplified user interface, one can dig down and explore and discover the structures of this virus. The next step we are thinking of is even less interaction heavy approach. And we have recently developed the concept, or I should say, David Kožil with colleagues has developed a concept called Molecumentaries, which he also generalizes to something which he terms Datamentaries, where we can create out of a model or a data which is annotated a certain documentary fully automatically. And I will show you how this can be done with some work in progress results. So the Datamentary Concept, and in our case now instantiated on molecular models, which is then called Molecumentary, starts with a 3D model, as we have the model of the HIV or SARS-CoV-2, and this 3D model is a hierarchically built-up structure, geometric model, which each part of it has a certain name and maybe even an ID. And you can use this information in searching for more information on the web about this structure. And we call this a story foraging. So we have initially some hierarchical buildup of our structure, and we are searching for descriptions of individual building blocks out there, maybe through Wikipedia API, or maybe using some other more, even more curated domain repositories, which contain information about the particular structures which are contained in our model. And this can be done actually in real time during the exploration process. Through this foraging, we gain additional information about each node, but also we gain information about additional relations between these nodes. So how one structure relates to another one, for example, in the context of metabolism or signaling, etc. Once we have such a story graph, we can then perform a traversal. We can choose a traversal through the story graph, which will then which will be then a narrative of a particular datamentary. This datamentary will be then codified with a voiceover, so a synthesized text, which guides the viewer along the story between the nodes. And to this audio, of course, a linked 3D visualization is created as well. So that these are synchronized streams, which are then put together in the datamentary, in our case, Molecumentary. So I will focus on the datamentary synthesis in the later slides, but I would like to show you one example of it, which is showing you the structure of the SARS-CoV-2 again.
Coronavirus disease 2019 COVID-19 is an infectious disease caused by severe acute respiratory syndrome coronavirus 2 SARS-CoV-2. It was first identified in December 2019 in Wuhan, Hubei, China, and has resulted in an ongoing pandemic.
So the narrative which you hear here has been taken from the Wikipedia API.
After focusing on COVID,
And on the right side, you can see how we are traversing the hierarchy. And always when we are traversing, you can see, you can hear a certain transitional text which leads you from one visualization target to another one.
Domain, guanine and others. After focusing on interior, we can see adenine. Adenine A, AID, is a nucleobase, a purine derivative. It is one of the 4 nucleobases in the nucleic acid of DNA that are represented by the letters GCAT. In the following step we're going to focus on nucleoprotein, C-terminal domain.
Okay, for the sake of time, I will fast forward here. You can see here the gist of idea, I hope. And so in our Datamentary synthesis, the visual part, we need to focus on several things. One is the camera settings and the camera transitions, where we are moving between more contextual views of a certain hierarchical level to a more detailed views, as can be seen on the left side. And we are performing various types of camera transitions. One is a zooming in transition straight on. Another transition would be retargeting from one object to another. And the third motion, which you might have seen, is motion when we are focused on a certain element. We are not having a still image, but we are slowly orbiting around that structure, which gives it a much more natural and enjoyable way of understanding the structure. This is coupled with what we call sparsification, because these structures are spatially occluding each other, but thanks to the hierarchical organization and the knowledge of the complete geometry here, we are able to open up the structure in an animated fashion and show what we are interested in. So we are using a simple cut plane in this case, which is now allowing us to see the structures of interest. And I would like to share with you one interesting way how an author can actually interact with this. Imagine that an author has created such a 3D model, like, for example, this HIV embedded in the blood plasma proteins, as has been created by the Scripps Institute. And then we asked the Scripps Institute, David Putzel namely, to write a short description of this model. And this text is actually the user interaction or the creation of the narrative, because we are automatically finding the occurrences of various keywords which are identifying themselves in to those which are within the hierarchy, and then we are focusing on those in real time, so that we are audificating the text here, and at the same time we are creating a synchronized video to it. You can see it, the initial version of it.
This model includes an HIV-1 virion, capturing the virus as it is ready to infect cells in the immune system. The virus is surrounded by blood plasma. The model is built with CellPack, integrating many sources of information from structural biology and microscopy. The virus is surrounded by membrane. Several envelope proteins are embedded in the membrane. GP120, one of them, recognizes receptors on the surface of cells that HIV infects, and is the major target for antibodies that block HIV infection. Researchers are studying GP120 in the search for new approaches to creating an anti-HIV vaccine. Several viral enzymes are packaged inside the virus. Reverse transcriptase will create a DNA copy of the genome when the virus infects a cell. Integrase will splice this DNA copy into the cell's own genome, which then forces the cell to make many copies.
One needs to say that while there is one hierarchy in this model, there are several instances of one particular building element. So we always need to also make a choice which of these building elements, which of these instances are we focusing on when performing the narrative of the model. But the overall idea is that you have just an
matrix protein is
array,
found on the inner
you have
surface
just
of the
an
membrane.
annotated 3D model, you have
It
a
plays a
script
major
to
role
it,
during
and
binding of the
the
virus from the cell
narration,
surface by
the
making
video,
sure that everything is
or
packaged inside.
other forms of visualizations are
Capsid protein
created
assembles
fully
into a
automatically.
hexamer.
I should quickly conclude, but let me just briefly mention that we are now, I think, slowly ready to open the Pandora box of time and try to again connect to some metabolic or life cycle models of certain structures, like for example this mycoplasma or the minimal genome organisms which are developed at the Craig Institute. We are currently have started the discussion with them how our visualization can actually help in their research. And the second aspect which I would like to highlight, that what we are working in the future on, is to create molecular models directly out of the Cryo-ET scans. So here we have some very early results where we can fit the envelopes into the structures which have been identified through this deep learning process, and also the positionings of the spike proteins. So with this, we not only will have procedural approaches to model these kind of models, but also a data-driven ones, meaning that we will actually end up in data-specific models. And these then can be used for further utilization in simulations, for example. When talking about simulations, we are also working on simulating the microscopy itself for various purposes, because as we are able now to create models of various structures, we would also like to create simulations of microscopy thereof, and for various deep learning training purposes. For example, to improve the reconstruction, to find the missing wedge artifacts, to be able to improve the segmentation and expand it beyond the classification of viral particles. So this is in short what I wanted to share with you today, and I will be more than pleased to answer any of your questions. Thank you.